All right. Welcome to Cantor Global Healthcare Conference. I'm Pete Stavropoulos, biotech analyst with Cantor. With us, we have MindMed, a company we cover. And I'm pleased to introduce Rob Barrow, CEO, and Dan Karlin, CMO. So let's start with a brief introduction of yourselves and description of the company for those not familiar.
Yeah, Dan, you want to start us off?
Sure. Yeah. Dan Karlin, Chief Medical Officer at the company. I'm a psychiatrist by training, also boarded in addictions and clinical informatics, faculty at Tufts in the School of Medicine, developed drugs for Pfizer before I got into the world of digital medicine for a little while, and then found my way back to drug development at MindMed just under 5 years ago.
Yeah, thanks. And Rob Barrow, CEO of MindMed. I've been working in this field, and it's easy transition over into our development program and organization of developing psychedelics for psychiatric disorders for the last 7 or so years, and here at MindMed for the last 5. And we're incredibly excited about the potential of what this drug class can do, and trying to build an organization to fully capitalize on that opportunity and really deliver something to patients that have not had anything new in many decades.
All right. So 7 years, that's and MindMed or overall in psychedelic space. So
working in psychedelics, I think.
Yes.
Yeah,
All right.
yeah, exactly.
Just get your perspective on how things have actually shifted in the past, 4 or 5 years. So you probably started when it wasn't more mainstream, and now it's moved a little bit towards the mainstream. And so just curious to see and hear your perspective, in terms of physicians that you interact with, or have psychiatrists sort of warmed up to it. When you go to a conference, how was it 5 years ago versus it is today? And so just curious.
I think there's always been an intrigue and excitement generally with the field, and particularly in psychiatry. There's been so little, again, in so many decades, that psychiatrists have been quite enthusiastic about any novel mechanism. But over the last 5 years, the data is now maturing. I think there was so much enthusiasm and excitement, but it was unclear the quality of the evidence, or exactly what was going to be happening. And now, we've been very fortunate to produce some incredibly exciting data in generalized anxiety disorder, and others in the field have generated data in other indications as well. And so, just the foundation that is being built for, and the data that can be relied upon. I don't know, Dan, if you had other thoughts.
Yeah, I mean, I think that the general acceptance in psychiatry, the idea that there are going to be psychiatrists who want to participate in this ecosystem, just goes up seemingly by the day. In the world of trialists, so psychiatrists who run trial sites and serve as PIs, there's an increasing number of sites now that it can do these sorts of trials and can do them really competently. Many of them also have clinical care sites that they have started to think about preparing to conduct clinical care when drugs become available. And I think maybe the most, it's soft, but it's the most enthusiastic sign I've seen is that when I teach residents now, which I do at a couple of places, usually I start by asking people, what is it about psychiatry that got you here, and what do you want to do? And the answer in groups has progressively moved toward, I want to practice with psychedelics. That there are people who are getting into the field of psychiatry because these tools are on the horizon.
And so you've seen expansion of interventional psychiatry.
Yeah. So, I mean, the ability to practice interventional psychiatry is limited by the tools, right? The tools for interventional psychiatry are TMS and Spravato. And if you're going to work off label and probably cash pay, ketamine. If you're really brave, ECT, but that's mostly in hospital settings and you need anesthesia resources and things like this. So there was, I think, a brief bubble of clinics that had been sort of created to do psychedelic work, but they were too soon. So we saw a little flare up of these folks who sort of miscued their clinic builds. There is obviously a large number of clinics that are on the Spravato registry, a smaller number actually prescribing Spravato. And so there is this interventional psychiatry world that's emerged around Spravato. TMS continues to be sort of background noise, but I think more than anything, it's this anticipatory. What we didn't see with the Spravato launch was anticipatory Spravato clinic building. right? There was ketamine clinics, but it took them years to start to switch over. It took insurance coming online for them to switch over, them to be forced to switch over because they ran out of people willing to cash pay for a ketamine injection every week or whatever. And so the anticipation in the psychiatry community of saying, here is what I can build to time it with potential drug availability, not getting ahead of availability, and people thinking about, okay, in my private practice where I might have a few psychiatrists, some LICSWs or PsyDs doing psychotherapy, what would it look like if we decided to start to do psychedelic interventions here?
Well, thank you for that. But let's go on and focus on MM120, which is your candidate breakthrough designation for GAD and major depressive disorder. For those that may be newer to the story, just walk us through why 120 and what it as LSD actually is.
Yeah, so when we look at the long history, and these are drugs, the whole class has been around for almost 100 years now. There was a period of real interest in the medical use of this drug class, really up until the early 1970s, the Controlled Substance Act. LSD was clearly the most prominent drug during that time, the one that was most widely researched. And looking at that sort of foundation and building upon that, we thought there was real potential, not only because of the clinical performance of the drug, but actually the longer lasting dynamics, which we think actually fit very nicely into clinical care. Dan was describing the world that sort of emerged in response to Spravato, which I think has been something that has been perhaps over-indexed on at this point, where there's a treatment of a 2-hour treatment session as somehow optimal. And we looked at the dynamics of a treatment session that it's a day for a patient. Most of these drugs, regardless of how long they're in or out of the body, are a day for the patient. And so we looked at those dynamics and the history and the mechanism of action of LSD, which is quite distinct from many other compounds in the class, and felt incredibly excited about its potential to drive meaningful change in these disorders. And so we really took that. I think there was also historically it was an overlooked opportunity, in part because of its history, and data and high quality science should overcome any of that, and we're just not scared to embrace that concept and move forward.
So you've gotten into GAD first. Many of your competitors, MDD or TRD. Why this? What's the rationale for GAD?
I'll turn to Dan.
Yeah. So the history, Rob talked about the history of LSD and it historically being the molecule of greatest interest to psychiatry and medicine. So was anxiety. In the history of psychiatry preceding the current SRI era, for non-psychotic illness, for let's say neurotic illness, the focus had always been on anxiety. Like anxiety is the background condition of being a person in many ways. And anxiety and depression have these massively overlapping populations, so that the way we really conceive of the world when you get past DSM diagnosis is that people with neurotic distress have a background level of anxiety that may or may not be at the level of a disorder. Certainly in our case, we're taking care of people who are at the level of a disorder. And then there are episodes of breakthrough depression. So major depressive disorder is intrinsically an episodic illness. The episode, we've allowed for longer and longer episodes per the DSM, up to 2 years now, but 2 months used to be sort of the outer edge of what you'd consider a depressive episode, because when you get beyond that, you're not talking about an episodic illness anymore. You're talking about someone who has chronic dysthymia, which often has a highly anxious flavor to it as well. And so with these diagnoses overlapping so much, we can think about chronic background anxiety with breakthrough episodes of depression. All we're doing with GAD is saying the current treatments for MDD and GAD work better on those depressive episodes than they do on the background anxiety. We have a drug that we have every reason to believe, and now based on the evidence we've accumulated, we know works for that background anxiety. So it's the historical condition of interest. It is the cause of the background distress for most people. It's often the symptom cluster that's left when people talk about TRD. Often those are highly anxious people. And so we said, let's bring something to this huge group of people who don't have good options today. SRIs often exacerbate anxiety. So it's huge unmet need. No one else is doing it. Let's go to where we can do something that makes a difference for people who don't have other options. And at the same time, we have an MDD program because there are people who don't have that background anxiety, who have major depressive episodes. We need to develop evidence that we can help people who are in a major depressive episode. By getting both of them, we cover a massive swath of the population who have psychiatric diagnoses.
All right. So you you did conduct a phase 2b, and I believe you published it yesterday.
That's right.
All right. Congratulations on that. And can you just walk us through the trial design and the key efficacy outcomes?
Yeah, so the phase 2 study, one of the things that was really remarkable about the field is that there's been, I think, a lot of assumptions, and there have not been many studies that have fully characterized even what we would typically expect from any drug, is like things like dose response. We sought to do that with a phase 2 study. We did a 5-arm dose response study where we looked at doses of 25, 50, 100, and 200 micrograms of MM120 versus placebo. It allowed for, of course, an assessment of activity, observation of the clinical outcomes at each of those levels. It also gives some really interesting insights of performance of lower doses and answering questions of things like functional unblinding, which is this somewhat catch-all term that's been talked about quite a bit in the field. What we saw in the study after a single dose of MM120, and this is with no concurrent therapy or psychotherapy, we saw a 12-week durable effect that was observable as soon as it could be measured. So on the HAM-A at 1 week, because it's a 1-week recall instrument, on things like the Clinical Global Impression of the Severity Scale, as soon as the next day, saw dramatic drops in anxiety and disease severity scores that were durable for 12 weeks. At 12 weeks, we had about half of patients in remission after that single dose. This is just staggering clinical response. And all those were at the 100 microgram dose level, which is what we're taking forward into phase 3. From the primary analysis, we established a statistically significant dose response. We also saw things that mentioned about functional unblinding. We saw that even though everyone who received any level of drug, 25 up to 200, 90% or more of them correctly guessed they were on drug. So everyone was functionally unblinded. The 2 low-dose groups didn't separate at all from placebo, and the 2 high-groups did, which one, I think, reasonably can conclude from that functional unblinding cannot explain the outcomes we observed, which really helped with our evidence and allowed us to think about a streamlined program in phase 3.
Okay. When you look at the effect size, how does that sort of compare to what you see with other approved drugs in GAD?
Yeah, there are very few drugs approved in generalized anxiety disorder, but those that are SRIs and benzodiazepines, primarily fairly modest effects. The standardized effect size is like Cohen's D of somewhere around 0.36, 0.38 for those drug classes. What we saw is that is an effect size of greater, more than double that, 0.88 at week 4, 0.81 at week 12. So more than double the effect size. And that was over an abnormally large placebo. That was a remarkable finding from that study as well. Is about a third of the patients who received placebo incorrectly thought that they were getting drug, presumably because there were lower levels of drug in the study. That was more likely. But that drove about a 14-point drop in the placebo group from baseline. The historical average for GAD would be more like a 9 or 10-point drop in the placebo arm. So we were able to exceed by 7.7 units from points on the scale, able to exceed an already large placebo. And when we think about the field, there's been concerns about nocebo effects and studies where there's just absolutely no placebo response. That obviously creates some questions about, about the magnitude of effect that is then observed or reported. We were so excited to see a large placebo response to sort of prove out the integrity of the study and study design, and then still exceed it by an effect size that's more than double the standard of care.
Right. Were these patients, this is a monotherapy, correct? These patients were not allowed to be on any alternative?
Correct. So patients who come in the study on some sort of pharmacotherapy are washed out from that before they're allowed entry in the study. So single dose in the absence of any other medications.
Right. And is it same for in the phase 3 studies?
Phase 3 studies are almost an exact replica. Obviously, we don't have 5 arms. We have 2 arms in one of the studies and 3 arms in the second study. But virtually everything, you know, all the fundamental design elements, the population, everything about the study is intended to be a replication of what we did in phase 2. And really, you know, we started our program designing a phase 3 study, but doing it in a dose response, to explore dose response. So the level of change between phase 2 and 3 has been pretty minimal, which is certainly gives us more confidence in potential there.
Okay. You know, what learnings from the phase 2B, you know, did you implement? I know you said that they're almost identical, but are there any learnings that you implement to help increase the probability of success of these studies?
Yeah, we certainly continue to streamline things, and maybe Dan could talk about some of the specifics there.
I mean, when it comes to operationalizing these studies, we're always learning. We learn on a daily basis, and we watch our studies at that sort of granularity. Like, we know moment to moment what's happening at sites and with individual participants. So everything that we learned from that level of supervision of the phase 2, we were able to start implementing in the phase 3. That starts with site selection. It's how we qualify the sites, how we engage with the sites, how we train the sites, how we train the DSMs, how we think about patient recruitment, how we think about patient flow through the study, and the timeline of those different steps. So the learnings are largely operational. There are, in a few cases, we learned that, say, an exclusion criteria wasn't excluding the people we hoped it would, so that we changed that to make sure that the thing about study protocols is that you're trying to take complex reality and document it in specified ways, pre-specified ways. And so that doesn't always work exactly how you think it will, which you learn from and adapt and make sure that you're getting the outcomes you want from the things that you put in a protocol.
So powering assumptions for these 2 studies?
They were designed to have 90% power to detect a 5-point difference, and both studies also include an interim sample size re-estimation. This is a blinded, no alpha spin, no inferential testing, but an analysis to allow us to maintain that 90% power if one of the nuisance parameters, dropout rate, standard deviation, if those are higher than we expect. So as we saw, 7.7 unit change was observed in phase 2. 5 points, we're planning on some sort of lower margin there, but we feel quite confident given the data we have to date and the likely outcomes there.
And the assumption, the dropout rate?
15%.
15%. And then, when you said you have the ability to upsize the study by what amount?
By up to 50%. We can go up to 50% larger than initially planned to maintain 90% power.
All right. So, there was a large placebo response in the phase 2B, like you mentioned, so how will you, what are you going to do to help reduce that placebo response in the phase 3s?
Fundamentally, things that drive placebo responses in any clinical trial are fairly common, I think consistent with what we saw in phase 2. The likelihood of a patient getting allocation to drug tends to increase placebo response. As is mentioned before, about a third of the patients who received placebo thought they were getting drug. That's probably likely to go down when we have fewer arms and fewer low doses that could confuse a patient and have them guessing. And so those features very well may reduce the placebo response. It's also by including in the studies, we have this 12-week double-blind parallel group period. Thereafter, patients continue in the study for an additional 40 weeks, where they have the opportunity for open-label treatment. That is also likely to increase patient retention. So in phase 2, patients who received placebo didn't feel better. They didn't get anything else from the trial. All they were asked to do is come back and, as you're reminded, they were off of the drugs that they had come into the study on. So a patient who was on an SRI, say, washed out, received placebo, it's a pretty big ask to ask them to stick around for 3 months when they have severe anxiety. And so they were the ones that were the most likely to leave. In phase 3, by having the extension period, we think that will likely drive up patient retention. That tends to have also an effect of numerically driving down placebo response, because the patients who are doing the worst were the most likely to leave the study, and their data from that point forward are treated replaced by those that did well enough to remain for the duration of the study. So even that feature, all of that, we would expect to potentially drive down a placebo response in phase 3. And as I mentioned, with phase 2 data that is so definitively established a dose response and shown a response over a robust placebo, we obviously feel the complementary designs we have across the program, the complementary results we would have, give us a lot of comfort, regardless of what that placebo response comes in as.
Okay. Can you talk a little bit about the open label study, open label extension? Sorry, and how many doses can they actually take, and what's the trigger?
So after the 12-week period, patients continue on to be observed. Give HAM-A, a primary outcome measure in the study, is given on a monthly basis. It's also given reflexively if screening criteria are met, the GAD-7 that would suggest a HAM-A is warranted. If patients have a HAM-A score of 16 or greater, they become eligible for retreatment. Obviously, they have to be deemed safe to be retreated as well. The real cutoff is a return of symptoms to a level of some moderate symptom severity. Patients can get up to 4 open label doses. And so we're really interested in that part of the study because it'll help us understand what real world retreatment dynamics could be like. When patients have up to 4 doses they can receive over the course of 9 months effectively. I think we have an expectation that most patients likely won't be using all 4 doses over that period, but it'll be quite informative as we think about and describing the retreatment dynamics and also looking at what happens upon subsequent retreatment. Obviously, we want to see that a subsequent retreatment also has some sort of clinical effect.
Okay. So what are you aiming for? 1 to 4 doses per year overall?
We think that something in that range would certainly be viable for this product, and there's going to be almost certainly a difference at an individual level. Some patients are going to take 1 dose and likely not take another dose for a long time. Some patients may need repeated dosing with some regularity, and that is perfectly okay. We think that's a viable model, and it allows for the appropriate treatment of patients when they need a treatment. It keeps them off, hopefully, taking certainly this drug every day. It won't be on hopefully a daily medication, and can have durable remission, durable clinical effects from the drug after just intermittent treatment, regardless of the frequency.
I guess one important question is, when it comes to psychedelics, is the length of treatment session. How long is the monitoring session for the phase 3 studies?
At phase 3, we keep patients in observation for 8 hours after administration. And think of it as a day. Patients come in the morning, they take the drug, they leave in the afternoon or evening. We start monitoring patients to be eligible to leave at hour 5. And so we have a structured, pre-specified checklist that we use to determine whether a patient would be safe to leave the clinic based on those criteria. And so we're just building a body of evidence to support a data-driven discussion around what is the appropriate length for an individual on average, at the low end, at the high end, all these sort of characteristics, which would be useful to inform its use in the real world.
Okay. And how many monitors per session?
So we require 1 monitor to be in the room. This is in line with FDA's guidance. We have the second monitor who's typically watching by video, remote from the room, watching by video. Their patients are under observation for the duration of the day. It's important though, but I think that dynamic and those standards sort of emerged primarily from a different drug with a completely different mechanism, MDMA for post-traumatic stress disorder. Very much a different treatment dynamic than what we see with LSD and the classic serotonergic psychedelics. So we've gone to great lengths too to document what is happening and to understand the dynamics of care for the dosing session monitors. And really what we find is that the folks sitting in the room with patients aren't doing much. Patients may need help going to the bathroom or want a snack or a drink of water during the day. But unlike the assisted psychotherapy models that had been developed previously, patients is largely internal. They're largely kind of quiet, laying on a couch and spending the day presumably thinking. And I think that dynamic has allowed us to really think and again, generate data to try to make arguments for what the appropriate level of monitoring should be, right? Any burden on delivery is going to limit access in the real world. And so I agree, I think be really thoughtful if we're going to put artificial restrictions on who could benefit from the drugs.
Or will the agency be thoughtful?
We've had great conversation with the agency throughout the development program on a number of topics, including this one. I think, I've over the course of working in this field, have already seen the agency take steps to move away from where they started. And so, and most of that was not even with data in hand, right? So as we've constructed arguments, what we tend to do is go in and say, look, this is the thing we're trying to solve. Here's how we're going to go about it. Here are the data we intend to generate to construct a rationale, and data wins the day. And so we and hopefully they will follow the data that we generate.
Okay. So, we're running out of time, but last sort of question. I mean, there's a lot more to go through. We think your other program, MDD, just we'll end with, if we're sitting here about a year from now, what would you like to say your key accomplishments have been?
Yeah, well, and of the 3 phase 3 studies we have running right now, we've got the first will read out in the first half of next year, and the other 2 will read out in the second half of next year. So we hopefully it's September. So hopefully sitting here in a year, we'll have a lot of data to be talking about together.
Awesome. Well, thank you very much for attending the conference.
Thanks so much, Pete.
And look forward to watching the progress and data in 1H26.
That's where it started.
All right. Thank you.
Thanks.
Thank you.